Brevity is the soul of wit. Here is a very famous quotation from Hamlet. Polonius, so imagine I am Polonius.
You are Polonius.
I am Polonius. The character who delivers these words means that speech and writing, to be efficient, to be witty, should be short. These words, so brevity is the soul of wit. Thank you very much. So these words mean that clever people can express what they wish to say in few words. They mean that being long-winded and verbose is the enemy of intellect. The words wit and wisdom both derive from a Germanic word meaning to know. Wissen. Pour ceux qui font nul. Wissen.
Good job.
Wit, wisdom. Yet. On devait le faire plus vite, hein, mais là, du coup.
Ralentis, on ralentit.
C'est pas waiting for Godot, c'est waiting for Claire. Okay. What is deeply comic in Hamlet and ironical is that these words on brevity are delivered by the most talkative character, Polonius. That is to say, by the one who is precisely incapable of being short and concise.
And we really wanted to respect the spirit of this event. We were going to be concise. We wanted to just warmly welcome you all here in Saint-Charles for this 3-minute thesis competition, which, as specialists in Shakespeare studies, we will consider today as a very Shakespearean event. And a Shakespearean experience as well. As vice presidents in charge of research at the University Paul Valéry, we are very happy to host this year's regional final and to open the event on behalf of the president of the university, Anne Fraisse, who is in Paris today and who could not attend the event here today. And as specialists of Shakespeare as well, we will just invite all the doctoral students here to be as clear as is the summer's sun.
An expression from Henry V, another Shakespeare play.
And to use the delightful measures
mentioned by Richard III.
But brief let us be.
This is a quote from Hamlet.
To every contestant we say, heaven and thee good fortune.
A quote from The Merry Wives of Windsor. Then, very common words. Let them begin.
That's from Romeo and Juliet. Thank you very much.
And welcome to Claire.
Yeah, just on time.
Ever since beginning my PhD journey, my parents keep asking me, have you discovered a treatment for cancer yet? Me? Not really. In the world of cancer, many challenges affect the efficacy of current treatments, and this complexity amplifies when dealing with glioblastoma, the most feared enemy of the brain, where survival spans around 15 months only. Let me tell you the story of glioblastoma. It all begins with a few mutants, cancer cells, multiplying continuously to form an army of invaders. This army expands in the brain, disrupting its vital functions. Surgeons might enter the fray, removing maximum of the army, followed by bombardments of radiotherapy and chemotherapy. Yet, the battle is far from over, as some invaders survive, forming a larger tumor by claiming new territories. During my PhD, I discovered one strategy of the enemy. Glioblastoma invaders are taking supplies secretly from normal cells. Their choice of supplies? Energy drinks. But not the kind you're thinking of. These are mitochondria. Beyond their role as sources of energy in the cells, they have other effects. In our lab, we're diving deep into how these mitochondria change cancer cell behavior. And my approach, I introduce mitochondria from healthy cells into cancer cells and observe them within a 3D mini tumor called organoids. And what I've discovered is staggering. Glioblastoma cells harboring these extra mitochondria produce collagen. It's like the web of Spider-Man. Once released, it enables the cells to migrate, conquer new territories, making the tumor much more challenging to defeat. So where do we go from here? My aim is to inhibit this collagen production, effectively marking the boundaries of the tumor. And this strategy would enhance the precision of targeted therapies. So, to my parents wondering about a cure for cancer, we're not there yet for glioblastoma, but step by step and discovery by discovery, we're inching closer, one mitochondrial secret at a time.
My grandpa used to have this little organizer in which his pills were sorted so that he wouldn't mix them up. There were several beta blockers against high blood pressure and phenodol against his asthma. He was also suffering from Parkinson's disease, which would add another 5 to 7 drugs to the list. You may be wondering, Rita, why are you telling us all this? Well, all those drugs my grandpa used to take were targeting a specific family of proteins in his body. This protein family is called G-protein-coupled receptors. In fact, 1/3 of all drugs on the market today target those receptors. However, many of the drugs come with side effects. Others become less effective when taken every day and over a long period of time. But to improve drugs targeting G-protein-coupled receptors, it is crucial to better understand how those receptors work. We know that G-protein-coupled receptors sit on the surface of almost every cell in our body. Their job is to transmit messages from the outside to the inside of the cell through a series of different motions. You can almost think of a G-protein-coupled receptor as a college student. Let's call him Tommy, playing a game of charade with his friends. Tommy receives a card with a phrase on it. He then executes a series of different motions, and as he's very good at the game, his friends understand what Tommy wants to tell them. How does Tommy decide what motions are the best to convey his message? We don't know. Similarly, we do not entirely understand how G-protein-coupled receptors convert a message into motion to transmit it from the outside to the inside of the cell. And that is what I intend to find out in my PhD research. I developed a computational pipeline that allows us to simulate in detail the motion a G-protein-coupled receptor executes after it receives a message to transmit. My pipeline gives us many three-dimensional models, which we can use to find drugs that act on the receptor and either block, accelerate, or modify its motion. This way, we can correct faulty messages that are sent to the cells in our body in case of a disease. I hope that my computer simulations will help us to find more efficient drugs against a wide spectrum of diseases, so that in the future, when one of us suffers of one or the other medical condition, we will have better drugs in our pill organizers than my grandpa did.
Welcome to the amazing metropolis within our cells, a city where millions, nay billions of molecules live. Inside, DNA turns into RNA, RNA into proteins, and they all live there to do their jobs. Imagine that you live in that kind of city, walking on the street or sitting in a bus, bumping into millions of people you don't know every day. There, you also have your loved ones, your friends, your families, people you just love being with. Imagine that because of the size of the city, you can't see them. So you all choose that one place to meet, where the moment you step in, you no longer have to work. You just sit there with your people. In my thesis, I work on germ granules. They are cell compartments that allow us to stay fertile. They allow our species to survive. And they are the meeting place where the RNA escape the cell, find their partners, and give us proteins to have sperms and ovules. I want to know exactly how the shape of these granules affect their function. The problem is, these molecules are very tiny. So I use a super resolution microscope and I color the RNA to look at them one by one, to know exactly where, when, and how they give us proteins. Put simply, in my hands, when the RNA give proteins, they light up, they shine, and I record this change. You know, lab work is tough, but sometimes you get to see things that no one in history has seen before. One late night in the lab, I found one of these things. I saw that germ granules have a unique structure. They look like donuts, and the RNA are just like little sprinkles, some inside and some outside. I found that when the RNA step inside the granule, they hug each other and sit there. But the moment they step out, they need to start working again and giving us proteins. And if we lose this structure, if we lose this meeting place, we lose our ability to reproduce. Understanding how the structure and the function affect each other will change the way we look at everything in biology. Because these unique structures, they are not just specific to germ granules. We can find them in every corner of every cell of all our bodies. And they control everything.
I want you to think about the last time you were in a classroom learning a foreign language. After many years of lessons, did you feel ready to go out into the world and speak that language? Well, I didn't. I spent years learning English and French in my country, Colombia, where everyone speaks Spanish. But I had a chance to travel to the United States and France, and I realized that interacting with native speakers helped me a lot to gain fluency. So it would be ideal, right? Just to travel abroad to learn the language. But in reality, most learners can't afford to study abroad. The option that they usually have is to study in a classroom setting. Then I started teaching Spanish at the university in France, and I realized that students spend years learning foreign languages in schools too. But when they start the university, they report that they're not able to speak them in real life. So I decided to find a solution to this problem, and I chose the development of speaking skills as the main subject of my research. First, I asked myself, what can I do to get my students to practice with Spanish speakers? I knew that in Colombia and France, most students don't have native speakers to practice with. So why not help them both? But how? Well, during the COVID-19 pandemic, hundreds of tools to communicate with others were developed. So in my research, I analyzed those tools and how they can be used to foster interaction between learners. So in order to do this, I've designed a telecollaborative blended course. This means that in my class, some lessons are face-to-face with me teaching the students in a classroom, and other lessons are online with my students practicing with native speakers. So in my course, my students who are learning Spanish in a university in France are in contact with Spanish speakers who are learning French in a Colombian university. The online exchanges are usually in pairs, but at the end of the course, we have a session all together. And this is a picture that we took the last time it happened. So I've been testing this course for 3 years now with different students each year. And the results show that after the course, 86% of the students feel more confident speaking Spanish. Besides, the pre-test and post-test used to determine their level show that they have gained fluency. So now I'm certain that this teaching method works. The next step is to apply to other contexts and other languages, because this method is another way of getting the advantages of traveling without the cost of it. So what are we waiting for? Let's try. It's free.
Have you ever had a delicious pizza? Very hot, with a lot of melted cheese. You are salivating, and when you take a bite, you feel it's tasty. This flavor of deliciousness is a real taste, considered as one of the 5 tastes. It's known as umami, a Japanese term that literally means delicious in Japanese. And umami is mediated by a molecule, glutamate, which is the molecule in our brain for transmitting messages along roads that are neurons. In each of our brains, we have 100 billion of these neurons, nearly the same number as stars in the Milky Way, in which glutamate moves. To prevent traffic jam on this neural road, we have synapses. And synapses are as checkpoints when we find the receptor of the glutamate that have one job only: collect the glutamate as a message and transmit the information to the right place. This family of receptor is divided into 8 members. And I'm looking for the 7th number of this family, because they are like blocks. They are associated 2 by 2 to be functional. And the number 7 glutamate receptor, it can lead to diseases if it's poorly regulated, like addiction. Then you eat too much of this delicious pizza without moderation. But how can we develop treatment against diseases like addiction without knowing exactly where these receptors are? This is where my thesis comes in. I do this in 2 steps. First, I develop tools to study the behavior of the receptor. Secondly, I use it to localize the receptor in the brain. I'm like an inspector who is trying to find someone based on a fingerprint. How do I do this? I study my glutamate receptor outside the brain with methods involving fluorescence energy transfer. A simple yet brilliant concept. I put a green light on in the left hand of the receptor and a red light off in the right hand. Then, when you give synthetic molecule to the receptor, the hands come together to receive it. The energy of the green light is transferred to the red light. Red light on equal message is taken. So far, I have identified molecules that specifically react with our receptor. And these fingerprints will be tested in different regions of the brain. So that's the path I take to paint the map. And this map allows me to reach the stars that contain our glutamate receptor in our magnificent Milky Way.
Would you believe me if I told you that an organism smaller than your eyelash is being used to discover new treatments for cancer? Caenorhabditis elegans, or C. elegans, is a tiny, simple worm that we study to learn more about larger, complex organisms like humans. You might be thinking, no way, these creatures don't look like us. But we're alike in invisible ways. We have 60% common genes. However, our similar genes operate differently. For example, gene RAS can be found in C. elegans, where it controls organ formation, while in humans it controls cell divisions. Imagine RAS as a gardener looking after a blueberry field. If many gardeners are working in the same field, they will produce an abundance of blueberries. But a field without gardeners remains empty. In C. elegans, RAS controls the formation of one reproductive organ. If RAS is activated more than usual, the excessive RAS proteins lead to the formation of many abnormal blueberries all over the worm. However, in humans, genetic modifications in RAS result in fast, uncontrolled growth of cells, which could form a mass called tumor. In fact, RAS activation is reported in a type of leukemia called AML, a cancer that happens when certain blood cells multiply quickly and uncontrollably. Current chemotherapy for AML hasn't changed for decades, and patients are developing resistance to it. My PhD is about the discovery of new drugs that could cure AML. I use a molecule called imicaline, created by our chemists, and as a biologist, I study its anti-cancer effect by testing it on cancer cells and C. elegans. Moreover, since C. elegans is an invertebrate that can't feel pain, so it allows us to reduce animal testing and suffering. After I treated the worms with imicaline, I found that the blueberry shapes disappeared. So this molecule is inhibiting RAS and controlling the abnormal organ formation in C. elegans. Then I wondered, could imicaline also inhibit RAS activation responsible for cancer in humans? Indeed, I discovered that imicaline put a stop to the rapid multiplication of cancer cells from AML patients and lowered the activity of proteins influenced by RAS. I want you to think about all the other health problems related to genetic changes that we have in common with C. elegans. And imagine how, one day, this tiny yet remarkable worm could be part of the solution to curing those diseases. Now, do you believe me when I say that I'm using a creature smaller than an eyelash to discover new treatments for cancer?
Dear jury, okay, this is very hard, really very hard. The level was extraordinary. And when I say extraordinary, I was completely bluffed by the level of everybody, myself and the entire jury. We all were very, very impressed. And I'd like to begin with a big round of applause. And if you will, I'd like to extend that big round of applause to your tutors, to your colleagues, to your friends. I'm quite sure you've repeated that with your friends and family. So can we applaud all of those who are behind the scenes? You were the behind the scenes people in this story, so thank you to you. As I say, and we all agreed on this, the level is very, very high. So when we were together trying to find, because we had to come up with one name, which is very hard, we were going into intricate detail because all of you, each and every one of you, were able to stand up here with self-assurance, with absolute clarity in your words, with a delivery that was really very good. You really caught our attention, kept it, held it to the last second, which is exactly how it should be. And all of you did that. Your English is great, obviously, very impressive. You come from different parts of the globe and all of you master English very, very well. So all of those boxes were ticked for everybody, okay? That's really what we want to get across. I think in the name of the jury, that's probably our number one message here. But as any jury, we had to make a choice. And that was, well, it was very, very difficult. We were more or less, let me say, in agreement, but there were some discrepancies. So, you know, we had to come up with one name, but some of us preferred one rather than another. And so I think that should reassure everybody that it wasn't an absolutely obvious choice that jumped out and hit us, okay? I'm now going to come to the name. I'm trying to stretch it out, okay, because it's hard. really, really hard. But what I want to say is, to all of you, before I give the name, keep your style. You all have a very individual style, each one of you, okay? Keep that style. Don't morph into something that is like everybody else. Stay how you are, because that's one of the things that was particularly pleasant, was that each character, each personality was displayed. Okay, so please keep that. Don't try and become someone that you're not. All right, that's probably a personal message thrown out to you. And at this point, I'm now going to have to say that the jury, with my dear friends here, we all decided to give the prize to Ali Haddad. Well done. Speech? Speech!
Did not expect it. First, thanks to the members of the jury, the École Doctorale for the Collège Doctoral for organizing everything. I'm losing my words. I would first like to thank Patrick and Marianne for all the training they did. We wouldn't have been at this level without them. And my colleagues, all 5 for you were amazing. I would never think about myself.
Really.
I would like to thank my team and everybody at the IGH as well, because they're my friends in Lebanon and Noor as well. Thank you.